Hello students, welcome to Raaonline, and the chapter for this session is Reproduction in Flowering Plant, NCERT 12th class book, chapter number 2. And this chapter basically deals with the embryology of angiospermic plant. This chapter is basically related with the embryology of angiospermic plant. In plant embryology section, first I want to introduce a person, that is late Panchanan Maheshwari. Late P. Maheshwari. Father of plant embryology. Father of plant embryology in India. And he wrote 2 books. An introduction into the embryology of angiosperm. Into the embryology of angiosperms. This is one book. Book number 2, Recent Advances in Embryology of Angiosperm. And Mr. Maheshwari was related with the Delhi University. He is related with the Delhi University. He was a renowned embryologist of our country. In plant, for the formation of new generations, that is, progeny of a plant depends on flower. That is, if flower is there, then there is a possibility of a next generation. This is for sexual reproduction only. This line is actually for sexual reproduction only. Mr. Camerarius discovered reproductive structure in plant. Camerarius discovered sex organs in plant. And flower is a reproductive structure of an angiospermic plant. Flower is reproductive structure of angiospermic plant. And these 2 whorls of flower, that is stamen and second is carpel. These 2 are enough for the production of new plant. That is, these 2 are concerned with the formation of fruits and seeds, and those seeds, if they shed, they give new plant. So Camerarius discovered a reproductive structure in plants, and flower is reproductive structure. Flower is a reproductive structure of an angiospermic plant. In plant, a bud appears in the axis of a leaf. This is a bud. This is bud which appear in the axis of a leaf. If it is floral bud, if this bud is floral bud, then it opens and develop into a new flower. So the opening of floral bud is known as anthesis. This is the answer of question number 1. First of all, appearance of floral bud on plant, then this floral bud gradually opens into a flower. The phenomena is termed as anthesis. In flower, question number 2, in flower there are 4 whorls. There are 4 whorls in a flower. Out of these 4 whorls, 2 whorls are reproductive. Out of these 4 whorls, the 2 whorls are reproductive. One is androecium and other whorl is gynoecium. Total 4 whorls are there in a flower, but only 2 whorls are reproductive, and these are the 2 reproductive whorls of flower, androecium and gynoecium. Androecium is termed as male reproductive whorl. Gynoecium is termed as female reproductive whorl. Androecium is fertile whorl. Because it is concerned with microsporogenesis. The whorl related with the microspore formation is androecium, and the whorl which is concerned with the megaspore formation or megasporogenesis, that is gynoecium. So these 2 whorls are fertile. Out of 4 whorls, 2 whorls of flowers are reproductive. Male reproductive whorl androecium related with the microsporogenesis, that is formation of microspore, and whorl number 2, it is concerned with the megasporogenesis, that is formation of megaspore. Come to androecium. The units of androecium are called stamen. Units of androecium are known as stamen. Androecium, male reproductive whorl. Androecium is male reproductive whorl. Its units are known as stamen. Its units are called stamen. And in stamen there are 2 parts, anther and filament. This anther has 2 lobes, lobe 1 and lobe 2. Anther is bilobed, and these 2 lobes are joined together by means of connective. This is connective which joins 2 lobes, and each lobe is with 2 sporangia. Each lobe of anther has 2 microsporangia. And within microsporangium, microsporogenesis takes place. In microsporangium, process of microsporogenesis takes place. Microsporogenesis takes place. In which structure? That is in anther. So formation and differentiation of pollen grain, because pollen grains are the microspores. Pollen grains are actually microspores. They are produced in anther by the process of microsporogenesis. So microsporogenesis takes place in flower, in androecium stamen, in stamen anther. Anther has 2 lobes and each lobe with 2 microsporangia, and within microsporangium microsporogenesis takes place, results in the formation of pollen grain or microspore. So the answer 2 is right here. Second answer is right. Megasporogenesis takes place in gynoecium, carpel, that is swollen base is ovary, within ovary ovule is present. In ovule, megaspore mother cells, megasporangium is there and that forms megaspores. So megasporogenesis takes place in ovule. Megasporogenesis takes place in ovule, nucellus of ovule. This process in ovule, whereas in anther, microsporogenesis. Meiosis takes place in sexually reproducing organism. Question number 3. Meiosis takes place in sexually reproducing organism, and meiosis takes place at one particular event in a particular cell, not in all cell. So meiosis in case of flower, takes place in 2 structure, one stamen and within stamen anther, and in anther microsporangium, and in microsporangium microspore mother cell. This is microspore mother cell. Microspore mother cell diploid. It divides by meiosis to produce microspore. This is case of stamen. In gynoecium, second structure in which meiosis is seen, gynoecium, units carpel or pistil. There are 3 parts of a carpel or pistil. This swollen base is known as ovary, and within ovary ovule is present, and ovule megaspore mother cell is present. Megaspore mother cell is found, and that cell divide by meiosis to form megaspores. That is, the answer is quite clear. That is, in any angiospermic plant, meiosis takes place at the time of spore formation, and spores are representative of gametophyte. Spores represents gametophyte. Spores are representative of gametophytic generation. Spores represents gametophyte. It means any angiospermic plant perform meiosis when spores are produced. That is, from sporophyte, formation of gametophyte from sporophyte. Formation of gametophyte from sporophyte, then meiosis is seen, and there are 2 structures in which seen. That one is stamen, the other one is carpel. Spores are representative of gametophytic generation. Question number 4. Diathecous anther, in which number of sporangia are asked and in each lobe. So diathecous anther means anther with 2 lobes. This is a stamen. Its swollen portion is anther. This is a stamen actually. This structure is stamen. and its upper swollen structure is anther. Upper swollen structure is anther. In this anther there are two lobes, that's what is dithecus. There are two lobes of anther, that's what is called dithecus. So anther is dithecus having two lobes, and in each lobe two microsporangia are there. Each lobe with two microsporangia. Each lobe has two microsporangia. Now answer is now quite clear. That is, total number of sporangia in anther. Total number of sporangia in anther, that is 2 in and 2 in this one. So total 4 sporangia are there, and in each lobe 2. In each lobe 2. This is in this manner. This question is actually in this manner. So there are 4 microsporangia in anther and 2 in each lobe. 2 in each lobe. Question number 5. In a flowering plant, archisporium gives rise. Archisporium. When anther starts its development, see here, when anther starts its development, initially it appears like a oval-like outgrowth, and after some times it becomes rectangular. It becomes rectangular, and the outermost layer is epidermis, and just below the epidermis archisporial cell is there. This is archisporium. This is archisporium. So anther has actually 4 wall layers. Anther actually has 4 wall layers. Number 1, epidermis. It is present since birth, since the birth of anther. Other layers are middle layer, endothecium next to epidermis. Endothecium is present. Then middle layers are present, 2 or 3. 2 or 3 middle layers are present, and the innermost layer is tapetum. Innermost layer is tapetum. Except epidermis, remaining 3 layers, they are responsible, they are produced by the archisporial cell. Archisporial or archisporium tissue. So here the answer is 1 tapetum and a sporogenous cell. Only the wall of sporangium is wrong. Wall of sporangium is wrong. Except epidermis, all the tissue. Except epidermis, all the tissue. So both wall plus sporogenous. Here the answer is both wall and episporogenous tissue contributed by the sporangium. The only wall layer, that is epiderm, it is present since the birth of anther. Which of the following statement regarding the structure of microsporangium is correct? Microsporangium is generally surrounded by 4 wall layers. Yes, right. These are epidermis, endothecium, middle layer and tapetum. The first one is right. Second, outer 3 layers perform the function of protection and dehiscence. This line is given in the NCERT. That's what is right. Cells from tapetum undergo meiosis. This is wrong. Tapetal cell does not perform meiosis. Meiosis is seen in microspore mother cell or sporogenous cell. In microsporangium, let's say in microsporangium, sporogenous tissue is present inside the tapetum, just below the tapetum. Sporogenous tissue is present. This sporogenous tissue divides to form microspore mother cell. Microspore mother cell. These are diploid. These cells are diploid. And these cells divide by meiosis. These cells divide by meiosis to produce microspores or pollen grain. Microspores or pollen grain. So, actual cell in which the meiosis is seen, actual cells in which meiosis is seen within microsporangium is microspore mother cell, not tapetum. So, first and second correct, third is wrong. So, answer is first one, 1 and 2 are right, whereas third is wrong. In angiosperm, all 4 microspore tetrads are covered by a layer. See here, microspore mother cell. Microspore mother cell, it is diploid. This is microspore mother cell. What is the ploidy of microspore mother cell? That is diploid. This microspore mother cell divide by meiosis to produce 4 microspores. They are arranged generally in this manner in dicot. That is 3 at one plane and 1 is above. The group of 4 microspores is known as microspore tetrad or pollen tetrad. This microspore tetrad or pollen tetrad is covered by a layer initially. That is, all 4 microspores are covered by a layer. This is the question, and this layer is known as callose. This layer is known as callose. It's a callose covering. It's a callose covering. So, answer for this question is callose covering, and when anther matures, this wall ruptures and each microspore is free from one another. So, initially 4 microspores derived from microspore mother cell by meiosis, they are in a group known as tetrad, and this tetrad is also of different type. If 3 are in one plane and at one is on above, then this is tetrahedral tetrad. All 4 are in one line, linear tetrad, then this is the covering, callose covering. They may be arranged in this manner also, that is isobilateral tetrad. This is callose covering. This is common in monocot. This is common in dicot. Tetrahedral tetrad is common in dicot, and isobilateral tetrad is common in monocot. Then it may be T-shaped tetrad. This is T-shaped tetrad. So, these are the various type of tetrad, and this is the callose covering. This is the callose covering. But you should learn only these two because these are important. Tetrahedral tetrad is common in dicots, and one tetrad, that is isobilateral tetrad, this one is common in monocot. So, I am writing here, that is tetrahedral tetrad. Tetrahedral tetrad is common in dicots, dicot angiosperm, and isobilateral tetrad is common in monocot. Common in monocot. And what is tetrad? That is group of 4 microspore. Group of 4 microspore. They are covered by, then answer is callose covering. This is 7th question. Now 8th question, most resistant biological material that is related with the anther. Anther wall layer. This is one side of anther only. This is one side of anther only. This is layer number 1, that is epidermis. This is layer number 1, epidermis. Then layer number 2, that is endothecium. This is endothecium. This is endothecium. Then 2 middle layers are there. Then 2 middle layers are present. These are the middle layers which are concentric. Middle layers. These are concentric layer. And at least their number is 2. These are the 2 middle layers. Innermost layer is tapetum. Innermost layer is tapetum. This is the innermost layer, tapetum. Tapetal cells, multinucleate polyploid cells. Tapetal cells, multinucleate polyploid cells. And inside the tapetum, microspores are produced. Within this tapetum, these are the microspores. These are all microspores. And if we see the microspore, its outer covering is exine. Outermost covering of microspore is exine. This is exine of microspore. This is exine of microspore. And this exine has specific chemical known as sporopollenin, that is contributed by the exine of microspore. Exine of microspore. Exine of microspore or pollen grain. Exine of microspore or pollen grain has deposition of sporopollenin. It has deposition of sporopollenin. And this sporopollenin is contributed by tapetum. Tapetum contributes sporopollenin. Tapetum contributes sporopollenin. And it is highly resistant biological matter. It is kept under wax or lipid category, fat category, fatty fat and lipid like category, and highly resistant matter. That's why the answer is number 4. So here I have already described from where it is contributed, and which cells are responsible for its contribution, and it is deposited over. So it is seen over the exine. It is seen over the exine of pollen grain or microspore. Seen over the exine or microspore pollen grain. Question number 9. These are the statements and we have to choose the correct options. This is, we have to select the correct option from these one. We have to select the correct option. So outer one, this is a pollen grain. This is pollen grain and its outer covering is exine made up of sporopollenin. This is right. First one is right. Thin inner covering is known as entine. This is also right and made up of pectocellulose. First is right, second is right. And this is the nucleus of microspore. When this microspore nucleus divides, it forms one large nucleus, other one is small nucleus. The larger nucleus is vegetative nucleus and a smaller one is generative nucleus. Now from these two nuclei, two cells are formed. That is large vegetative cell and a small generative cell. The large one is vegetative cell and the small one is generative cell. It is generative cell, G cell I have written, small में, that is generative cell. This generative cell leaves periphery and floats inside the larger cell. This is larger cell, vegetative cell, and the floating one is generative cell. This is generative cell. So now see the options. Outer wall exine, right. Second one, inner thin pectocellulosic entine, right. Generative cell is bigger, this is wrong. And vegetative cell is small, this is wrong. So answer is 1 and 2 are correct. 1 and 2 are correct while 3 and 4 are false. Question number 10. Question number 10, that is, if we take the TS of anther, it looks like in this manner. If we take the TS of anther, it looks like in this manner. This is epidermis, then this is endothecium, second layer, then two middle layers, and the innermost layer of anther wall or microsporangium wall is tapetum. That is, innermost layer is tapetum. Tapetum is innermost layer. Tapetum is innermost layer. And tapetal cells are larger. They are multinucleate. Tapetal cells are multinucleate. They are rich in cytoplasm, dense cytoplasm. They have nutritive material also. And within the tapetum, sporopollenin containing bodies are present. Within tapetum, these are the sporopollenin containing bodies. These are tapetal cells and these are Abish bodies. And these Abish bodies are sporopollenin containing structure. These are sporopollenin containing structure and they contribute sporopollenin to outer wall of microspore or pollen grain, that is exine. This is exine, outer wall of microspore. Outer wall of microspore is known as exine and it is rich in sporopollenin. This is rich in sporopollenin that is contributed by the Abish bodies of tapetum. So Abish bodies of tapetum, they are concerned with the formation of sporopollenin. Question number 10. Answer for the question number 10 is sporopollenin. Pollen kit is actually oily sticky layer. What is pollen kit? This is insect pollinated pollen grain. Insect pollinated pollen grains are called entomophilous pollen grain. Entomophilous. They are called entomophilous pollen grain. Entomophilous pollen grains. That is carried out by insect. And on these pollen grains, oily sticky material is present. On entomophilous pollen grain, sticky material is present, that is known as pollen kit. That is known as pollen kit. And pollen kit material is also contributed by the tapetal cell, but not by the abish bodies. Abish bodies only contribute sporopollenin. And pollinia is a different structure, but pollen kit is found in insect pollinated pollen grain as oily sticky layer, outermost oily sticky layer. It is sticky, that's why it is easily, these pollen grains can easily adhere with the body of insect. Next, male gametophyte of angiosperm, monocot. Male gametophyte of angiosperm monocot is microspore, first cell of male gametophyte. First cell of male gametophyte is microspore or pollen grain. That is, from spores gametophytic generation starts. Gametophytic generation starts from spore. And angiosperm produce two types of spores, microspores and megaspores. Angiosperm produce two types of spores, microspores and megaspores. Microspores, first cell male gametophyte. First cell male gametophyte. Whereas megaspores, first cell female gametophyte. Female gametophyte. So answer is 3. Question number 12. See, direct question, that is study of pollen grain palynology. Study of pollen grain is known as palynology. And P.K.K. Nair, P.K.K. Nair, father of palynology, N-A-I-R can also be written. That is father of palynology in India. Famous palynologist of India, P.K.K. Nair. And study of pollen grain, that is palynology. Now, when pollen grain forms male gametophyte, this is pollen grain, initially single microspore with nucleus is there. This microspore or nucleus divides unequally to form two nuclei. This is vegetative nucleus, this is vegetative nucleus, and the smaller one is generative nucleus. The smaller one is generative nucleus. Larger is vegetative nucleus, a smaller one is generative nucleus. This vegetative nucleus forms A large cell known as vegetative cell, the smaller one is generative cell. This generative cell leaves periphery. This is two-celled stage of male gametophyte. Large cell is vegetative cell. The small cell which is floating within the larger cell is generative cell. This generative cell forms male gamete. This generative cell divides to produce two male gamete. When there is a division in generative cell by mitosis process produce two male gametes. These are the two male gametes. They are formed by generative cell. These are the two male gametes formed by generative cell. So come to the question now. If generative cell was destroyed by laser, then generative cell, if it is destroyed, then these two gametes will not be formed. These two gametes will not be formed. But normal pollen tube is still formed. Pollen tube is actually contributed by the vegetative cell. Pollen tube is actually contributed by the cell. To see the cell, this is vegetative cell. Generative cell is already destroyed. So vegetative cell, its inner wall intine protrudes and form pollen tube, because vegetative cell has no arm. Vegetative cell germinates to form pollen tube, but without gamete, but without male gamete, because male gametes are contributed by the generative cell. So the answer is now clear, that is vegetative cell is not damaged. This is not the, yes, this is the answer, because vegetative cell is normal cell, whereas generative cell is lost. Content of generative cell kills and stimulates pollen growth. This is wrong. Laser beam stimulates pollen growth. This is also wrong. And reason of emergence of pollen tube is not harmed. This is wrong. Vegetative cell is still there, so it is able to form male pollen tube. And which of the following is spermiogenic cell? That is gamete producing cell. Spermiogenic, the cell responsible for the production of male gamete. Cell responsible for the production of male gamete. Cell responsible for the production of male gamete cell responsible for the production of male gamete is known as a spermiogenic cell. And in the past question, I have already described the development of male gametophyte. That is, this is generative cell, this is generative cell, this is vegetative cell, this is vegetative cell, and this floating cell is generative cell. There is a mitosis in generative cell. There is a mitosis in generative cell results in the formation of male gamete. So mitosis in generative cell results in the formation of male gamete. Hence generative cell is spermiogenic. Generative cell is spermiogenic. And you also see that is this vegetative cell in future forms pollen tube. So pollen tube forming cells is known as siphonogenic cell. The cell which forms pollen tube is known as siphonogenic cell, and cell which forms male gamete is known as spermiogenic cell. So here the correct answer is, during the development of male gametophyte or at the time of germination of male gametophyte, generative cell will form male gametes. So it is spermiogenic cell, whereas siphon, that is tube, is formed by the vegetative cell. So vegetative cell is siphonogenic. Androgenic haploid. androgenic, that is pertaining to male. In that case, actually the word haploid is given. Wall is diploid, anther wall is diploid, so not possible. Tapetal layer of anther wall, it is also diploid, so not possible. Connective tissue is diploid, so not possible. The only option left, that is pollen grains are haploid. Pollen grains are haploid. If plants are raised from the pollen grain, they are called androgenic haploid. Plants raised from pollen grain, this is androgenic haploid, because ploidy of pollen grain is haploid.